central message that I was trying to deliver in the plenary lecture this year was about the value that you can distill from doing whole genome sequencing of cancers. So that is reading the entire genetic code of a human cancer. And the themes that came from there were about not just looking at driver causative mutations in cancer, but also use the many thousands of passenger mutations, things that we call signatures. They produce patterns in the genome. And the reason that's really valuable is that they give you insights into what's going wrong in the tumor as the cell's going from being a normal cell to a cancer cell, because these become targets for therapeutic intervention. So the central theme of the plenary lecture was the value of looking at whole genome sequences from breast cancers. Some of the key developments in our research was to not just define these mutation patterns, these signatures in the genome, but to also show how to use them clinically. So we've been able to identify that there are some patterns that tell us about specific DNA repair pathways that are going awry. And in the plenary lecture, I highlighted a couple of those DNA repair pathways, specifically mismatch repair deficiency, as well as HR, or homologous recombination repair deficiency. So we're trying to push the use or the utility of those mutation patterns, those mutational signatures, so that they become translated into clinical applications. So some of the key things that I'm hoping will come out in 2018 is that we will be able to marry together the genomics, the genetic features in tumors, together with clinical trials in a more systematic way and in a more comprehensive way as well. We can now do genomics in a scale that's unprecedented, but I don't think it's being used enough. So in my plenary lecture, I was really trying to sell the idea that actually we don't have to be scared about genomics. All the clinical trials going forward should absolutely be thinking about using the genomics that we can produce today, to not be afraid of it and to apply it in the clinical trial. So I'm hoping that in 2018 we're going to see more of these comprehensive combinations of genomics in clinical trials. Found some of the clinical trial talks where there's combinations of immune checkpoint inhibitors with various other treatments has been quite interesting. Clearly there are some trends, some fashions at the moment. But yeah, the increasing sort of use of an awareness of the basic biology underpinning why a tumor is abnormal, and using that context in clinical trials, I think we've sort of found that dotted around very many different sessions.